this webinar happen. He is backed by the organizing committee, which consists of Srimati Stuti Mohapatra of the same department, Suraj Ali of physics, Saminur Rahman of journalism mass communication, Bablu Shamanto of geology, who were his main back strength. Now, I would address my two eminent speakers who are gracing this webinar with their valuable time and effort and will be sharing their views related to the topic mentioned above. Dr. Shumit Sarkar, research biologist, Division of Neurotoxicology, FDA, that is US Foods and Drugs Administration in USA, and Professor Ashish Kumar Panigrahi, Pro Vice Chancellor of Bardhaman University, West Bengal. Now, Muralidhar Girls College has just crossed its 80th anniversary with a reputed and heritage legacy, but the Department of Zoology is pretty new. After a lot of hard work, the science wing of this college was strengthened first with the inception of the subject zoology in 2016, just 3 or 4 years back. It was then headed by Srimati Indrani Bhattacharya, who gave her very best to build it up from scratch. As she migrated to her new world, Dr. Surojit Shu took it up as in charge, was head of the department, and is till date surging forward, so I believe, gloriously, which soon I hope to be mutated into a honors course in the future. Hope that today's webinar paves the pathway, and I thank all of you for wishing absolute success for this seminar. Lastly, I wish to mention my ICT team, which is our back force and in-house effort, and whose valuable assistance had made this webinar come true. Thank you all once again, and happy webinar visiting. Over to you, Surojit.
Thank you, madam, for your kind words. Today we have two distinguished eminent speakers. In the first session, our first speaker is Dr. Shumit Sarkar, research biologist, Division of Neurotoxicology, Food and Drug Administration, USA. Dr. Sarkar received his doctorate in neuropharmacology and neuroscience from Nagpur University, Nagpur, India, in 2001. In 2000, Dr. Sarkar received further training during a postdoctoral tenure in the Division of Endocrinology and Metabolism at Tufts New England Medical Center, Boston. Later, at Indiana University, the incumbent studied emotional stress-induced cardiac regulation and fever to explore common hypothalamic origins and brainstem mechanisms, and found a common locus which is the key in thermoregulation, locomotion, and heart rate. Thereafter, Dr. Sarkar worked as a clinical research fellow in the Simon Cancer Research Center in Indianapolis for 1 year. And in 2008, Dr. Sarkar joined Boston Children's Hospital, Harvard Medical School as a research scientist to study the role of ER stress in inducing obesity and diabetes. In November 2008, Dr. Sarkar has joined at FDA, or you can say Food and Drug Administration. Dr. Sarkar's laboratory investigates the role of neurovascular elements, ER stress, and endothelial dysfunction in neurodegenerative disorder, with a special emphasis on Parkinson's and Alzheimer's disease using the rodent models and postmortem human brain samples. Dr. Sarkar is regarded as an expert in the field of Alzheimer's disease. Dr. Sarkar frequently served as an ad hoc reviewer for several prestigious peer-reviewed scientific journals, including Scientific Reports, Nature Communications, Alzheimer's and Dementia, Current Alzheimer's Research, Metabolic Brain Disease, Brain Pathology, Pharmacology, Biochemistry and Behavior, Neurotoxicology. Dr. Sarkar has served as a grant reviewer for Alzheimer's Research Association since 2012. He is also editor on international journals such as Metabolic Brain Disease current Alzheimer research. So now I would like to request Dr. Sarkar to deliver his speech. Over to you, Dr. Sarkar. Wait.
Sir, please unmute yourself.
Can you hear me now?
Okay.
Yes.
Yes, please.
Thanks for inviting me for this presentation, and it's an honor and privilege for me to speak in front of you guys. So let me see whether I can put my first slide. Now, can you see the first slide? Surajit, can you see the slide?
Just about to. Yeah. We are.
Okay, and can you hear me properly?
Visible and audible.
Absolutely fine.
Yeah. Okay, good. So one thing I would like to mention, let me make it pretty loud. Okay, can you hear and see the slide pretty easily?
Yeah.
Okay. I have a disclaimer to tell, because this is kind of regular and it's routine for us to put it, because this presentation is only mine, is not a formal dissemination of information by our organization or the agency, or any, it doesn't represent any position or policy. So whatever is coming out of my mouth is entirely mine. Nobody can hold responsible our organization. So without further ado, I would like to talk about today's presentation, which is nothing but neurovascular dysfunction and neurodegeneration in Alzheimer's disease. Some of the terms may be a little bit hard for some people, but I think some of the words will be very well explained in the coming video within a couple of minutes. So I will be presenting about three things. One is what is the central dogma about Alzheimer's disease, and what is the vascular dysfunction and neurodegeneration happen in Alzheimer's disease, and last but not the least, about my ongoing study, which entails about study of this vascular dysfunction in brain of transgenic rodent models, because we cannot do the clinical research because I'm not an MD. And their dietary impact and relevance to human Alzheimer's disease. So why I'm doing this Alzheimer research for the last 12 years? Because Alzheimer's disease is the fourth leading cause of death in the United States and the most common cause of adult onset dementia. And it actually affects around 35.2 million people worldwide. And that number is expected to rise about 65.7 million in 2030, according to Alzheimer's Disease International. And national direct and indirect monetary costs for caring for these people actually is about 100 billion annually in the United States. And not only that, nursing home costs for these patients currently about 50,000 per year and over 300 billion annually. Unfortunately, 66% of the Americans with Alzheimer's disease are women. So this is the video I'm going to show you. Just bear with me for a minute.
Alzheimer's disease was described for the first time in 1907 by the German psychiatrist Alois Alzheimer. In performing a histopathologic study of the brain of his patient August D., suffering from dementia, he brought to light the presence of two types of lesions in the brain: senile plaques and neurofibrillary tangles. He reached the conclusion of a distinct disease of the cerebral cortex. 100 years later, thanks to current scientific techniques, research has made a great leap in the understanding of the disease. We know that the brain is made up of neurons, and that these are interconnected to form a vast network. These connections, known as synapses, enable the transmission of information from one neuron to another. In Alzheimer's disease, 10 to 15 years before the appearance of the symptoms, two main lesions form in the brain: senile plaques composed of amyloid-beta protein, and neurofibrillary tangles, composed of tau protein. How is the senile plaque formed? On the surface of the neuron is a large protein called APP. Normally, APP is sectioned by enzymes on the surface of the neuron, and it frees a protein called amyloid beta. The amyloid beta protein is then cleared in the body. In the case of Alzheimer's disease,
Shurojit, you cannot see the video? Hello?
No,
The
not
video
able to
just
see the
started.
video.
The video just started when you just stopped it.
Okay, hold
Before
on.
that, it was not coming.
Because yesterday we presented, it was there. Let me,
Yes, yes.
unfortunate. Let me double check. One second. Can you see and hear now?
Yes.
Can you see here?
The visual has again gone, but sound is coming.
Alzheimer's disease was described for the first time in 1907 by the
Yes, it's
Can you-
coming. Yes,
It's
it's
coming
coming.
now?
Yeah,
Yes.
it's absolutely fine.
Alois Alzheimer. In performing a histopathologic study of the brain of his patient Auguste D, suffering from dementia, he brought to light the presence of two types of lesions in the brain, senile plaques and neurofibrillary tangles. He reached the conclusion of a distinct disease of the cerebral cortex. 100 years later, thanks to current scientific techniques, research has made a great leap in the understanding of the disease. We know that the brain is made up of neurons and that these are interconnected to form a vast network. These connections, known as synapses, enable the transmission of information from one neuron to another. In Alzheimer's disease, 10 to 15 years before the appearance of the symptoms, two main lesions form in the brain: senile plaques, composed of amyloid beta protein, and neurofibrillary tangles, composed of tau protein. How is the senile plaque formed? On the surface of the neuron is a large protein called APP. Normally, APP is sectioned by enzymes on the surface of the neuron, and it frees a protein called amyloid beta. The amyloid beta protein is then cleared in the body. In the case of Alzheimer's disease, there is an imbalance. The amyloid beta protein is no longer regulated and is found in too great a quantity. The proteins assemble to form indissoluble fibrils and create senile plaques. How are neurofibrillary tangles formed? When a neuron communicates with another, a signal goes from the body, known as soma, to the synapse to transfer the information. The signal passes through the skeleton of the neuron composed of microtubules. These microtubules are stabilized by normal tau protein. In Alzheimer's disease, tau protein becomes defective and detaches from the microtubules. Thus, the skeleton of the neuron dissociates as it is no longer maintained. Defective tau proteins then assemble to form filaments in the neuron. Without the skeleton, the neurons degenerate and connections between the neurons are lost. The abnormal accumulation of tau filaments in the neuron creates neurofibrillary tangles and eventually causes the death of the neuron. How do the two lesions spread throughout the brain? Neurofibrillary tangles and senile plaques do not follow the same pathway in the brain over time. Neurofibrillary tangles first develop in the region called the hippocampus, which is essential to memory and learning. They then reach the whole brain following a centrifugal movement. The process causes atrophy, which engenders global dysfunction. The progression of the lesions corresponds with the symptoms of the disease, which begin with memory problems, followed by problems of language, recognition, and incapacity to perform gestures. Senile plaques develop differently. They are initially observed in the cortex, secondly in the hippocampus, and then the senile plaques reach the whole brain following a centripetal movement. Their progression does not correspond to the symptoms of the disease. But numerous questions remain unanswered. We know that the presence of the two cerebral lesions is necessary to develop Alzheimer's disease, since one does not come without the other. But which lesion comes first? Neurofibrillary tangle or senile plaque? The answer is still under debate. Many clinical trials destined to reduce senile plaques in the brain have failed. In fact, reducing them is not sufficient to eradicate the disease. It has now been suggested that well before formation of senile plaques, smaller forms of amyloid beta called oligomers appear to be toxic for neurons, disturbing their communication when they fix onto synapses. It would appear that the toxic oligomers and their accumulation in senile plaques are at the origin of neurofibrillary tangles, which in their turn are responsible for symptoms. The relationship between amyloid beta protein and tau protein is still little understood. What is the exact sequence of molecular mechanisms leading to the development of dementia? What is the role of genetics and environmental risk factors in the appearance of the disease? Scientific research is essential in answering these questions. Thanks to researchers, Alzheimer's disease is better and better known in its complexity, and new avenues raise real hope for the eradication of this devastating disease.
Can you see the slide? Hello?
Yeah.
Yes.
Okay. Just to make sure, because when you do the transition, sometimes it becomes a little bit difficult for others. So the main hypothesis about this Alzheimer's disease, it became like apparent in 1992 when two scientists postulated that amyloid beta is the most important and causative agent in Alzheimer's disease pathology, and that neurofibrillary tangle, cell loss, and the other things ensues, such as, and also the direct result of this deposition. So what are the basic aspects or basic mechanism of this amyloid precursor protein and ultimately the A-beta? So this amyloid precursor protein is nothing but a type 1 membrane protein, which is sequentially cleaved, as you see in the figure on the left side, by two aspartic proteases to generate the A-beta. First, what happened, the beta secretase enzyme actually cuts this APP to create the N-terminus, and then these two fragments are produced, and then C99 and secretase beta ectodomain actually second this C99 cleaved by GABA secretase enzyme and to generate the C-terminus of this A-beta. So this A-beta actually is the toxic one. So how does it look in the Alzheimer's disease brain? Because the definition actually came from the person who actually dealt with this patient, whose name is Alzheimer. That's why this name came, Alzheimer's disease. As you can see on the right side, the brain section of a Alzheimer, as well as in the normal, you can see the difference between in the ventricle size, the ventricular enlargement, and the memory formation, the hippocampus area, it's been atrophy and also get reduced. At the bottom of the slide, you can see the most toxic part of this Alzheimer, disease, which is called 40 to 42 fragments. So, as I said in the also in the video, it showed that most importantly, two salient features we call plaques and the tangles. The plaques are the main, it's like a deposition, which is mostly, extracellular, and the intercellular is the tangles, and is pretty predominantly found in the cortex and hippocampus. So next, see what are the genetic factors that actually can increase this A-beta aggregation. Autosomal dominant familial AD, or we call it this FAD, is caused by mutation in 3 different genes, APP, presenilin 1 and 2, which are all integrally involved in A-beta production. FAD mutation led to accelerated accumulation of A-beta plaques and caused early onset dementia, which means this particular type of dementia, we call it genetic, and this version is cruel, and it can happen between 35 to 45 as well as 50. So, but duplication of these APP locus in chromosome 21 can also increase the A-beta production through increased gene dose and cause age-related dementia with brain parenchymal A-beta deposits, and which is actually apparent in Down syndrome, who have trisomy 21 and thus have an extra copy of this amyloid precursor protein, which actually develop the age-related amyloid plaque deposition. Remember, Down syndrome also have 2 different types. One is the age, in a very early age, and they can die. Another one is mild, which they can live a little bit longer. So a lot of studies going on about this particular Down syndrome patients having Alzheimer's disease. Fortunately, the FAD, or familial Alzheimer, is extremely rare, and the vast majority of these Alzheimer cases that we call it sporadic or SPAD. So genetics of this sporadic Alzheimer disease is very complex, and the strongest genetic risk factor is ApoE. What is exactly ApoE? Which is nothing but a glycoprotein which expressed mostly are produced by astrocytes in the brain and the liver and in the periphery, and important for lipid metabolism. So what does it do in terms of Alzheimer's disease pathology? Approximately 20 to 25% of the population carries this 1 copy of ApoE4, which increases the risk of Alzheimer's disease by 4-fold, whereas 2% of the population carries 2 E4 alleles, imparting at least 12-fold increased risk. Remember, this ApoE4 also a very, very risk factor is atherosclerosis, and it has been found that atherosclerosis patients also have more, preponderance of this Alzheimer's disease among these ApoE4 and atherosclerotic patients. So accelerated amyloid plaque accumulation is found in human ApoE carriers, as I said, and preclinical studies also showed that in ApoE can influence A-beta metabolism, with ApoE promoting amyloid aggregation and also deposition. So in the mice studies, they showed that reducing the ApoE levels genetically or with the antibody therapy can reduce the A-beta plaque and the disease pathology. Although the ApoE4 appears exert a variety of effects in the brain, strong modulator of A-beta pathology as well. As I said in my previous slides, the FAD, or familial Alzheimer's disease, this particular family in Colombia genetic mutation causing this Alzheimer's disease, and a lot of scientists are working on there, and there are a lot of studies going on, and among them is called Banner Health Research in conjugation with NIH and Genentech. They have poured a lot of money, like $50 million, to study of these people with this particular therapy, and the study results will be revealed pretty soon in next year, I guess. This particular version is very cruel version. It strikes in the early mid 40s or in late 30s, and the patients are in a very sad situation, extremely debilitating, because they cannot even take shower on their own or can even feed them by themselves. So it is a cruel version of the Alzheimer's disease. So how does this A-beta actually initiate the AD pathology? That's $1 million question. We all know that fibrillar plaques, oligomeric forms of A-beta, can mediate the further pathology in Alzheimer's. Various cytotoxic oligomeric, as you say in the video, the species has been identified in Alzheimer's disease patient's brain, lysates, can also exact, in vitro cell culture, we can see that it actually causes pathogenic effects, and also in the mouse brain. These oligomeric A-beta do not necessarily correlate temporally or spatially with the plaques. Some evidence suggests that the presence of these oligomeric A-beta predicts the presence of dementia more accurately than plaque burden. In human, actually, these oligomers appear to accumulate with age and are pretty correlated with the appearance of tau pathology. And the in vivo study, the mice which develops A-beta oligomers, but not the fibrillary plaques, also develop synaptic damage, inflammation, and ultimately cognitive impairment. So A-beta oligomer in vivo has shown that it can directly initiate the tau phosphorylation in vitro and in vitro. Remember, tau phosphorylation is a major causative part of the neurofibrillary tangle formation in the brain. At the molecular level, oligomeric Abeta can initiate tau pathology or tauopathy through the specific signaling mechanism. We call it SRC kinase FIN. And actually, ultimately, what it does, it induces the proteostasis, which can modify tau or activating specific tau targeting kinase. Abeta, on the other hand, can cause also large-scale alterations in the brain's ability to maintain proper protein quality control, eventually prompting aggregation of tau, and also sometimes another protein, which is synuclein, which is part of Parkinson's disease formation. But Lewy body dementia and Parkinson's disease also have some commonality with the Alzheimer's disease. What are the other factors? Age-related effects or comorbid pathology, which I'll deal the last slide when our recent Lancet studies and Lancet recommendation about it. Diabetes, atherosclerosis, head trauma, oxidative stress, inflammation, vascular factors, and also even sleep deprivation can influence the rate of Abeta aggregation and accumulation, and also accelerate the rate of Abeta triggering downstream pathology, thereby modulating the onset of dementia. So we have talked about the Abeta and the tau. So if Abeta is the initiator of AD pathology, what exactly can trigger the aggregation or accumulation of Abeta in the sporadic Alzheimer's disease? And how it is related to age? Plausible explanation is Abeta might accumulate in a constant and dependent manner throughout our life. It accumulates in certain brain regions, leading to excessive Abeta production, ultimately causing aggregation and seed formation, and then it propagates factors that enhance Abeta production or aggregation and suppress Abeta clearance, could contribute to this over a lifetime. I'm hearing a feedback. Can someone mute their phone? Impaired sleep-wake cycle could contribute to gradual Abeta accumulation by promoting a higher level of neuronal Abeta release and by disrupting normal diurnal oscillation in the extracellular Abeta levels. Sleep deprivation, there are studies going on saying that sleep deprivation can also impair the clearance of Abeta from our brain. And it has been found in the some in vivo preclinical studies that excessive sleep deprivation can cause more accumulation of Abeta in the brain. Age-related oxidative stress and nitrative stress can exert a gradual influence of Abeta production by altering secretase function. Age-related decline in the clearance of Abeta can cause also initial Abeta accumulation. Studies in humans recently suggest that diminished Abeta clearance has been found in Alzheimer's patients. Another key, but what is the discordant we found in the tau pathology and neurodegeneration? One key argument against the amyloid hypothesis is pretty poor correlation, because temporally and anatomically, Abeta plaque deposition, neuronal death, and clinical symptoms in Alzheimer's disease does not correlate very well. So neuroanatomically, fibrillar Abeta deposition occurs first, and most severely in regions such as the precuneus and frontal lobes, whereas the neuronal death begins and occurs mostly in entorhinal cortex and hippocampus, which actually has more neurofibrillary tangles than the plaque initially. So tau pathology correlates much more closely with the neuronal loss, both spatially and temporally, than amyloid plaques. Abeta pathology appears to begin in the cortex and spread inward, whereas tau pathology exhibits in the opposite direction. Familial Alzheimer's disease, which is driven by Abeta, should have a stronger anatomic correlation between Abeta deposition and neuronal loss, but that is not the case for sporadic Alzheimer's disease. Genetic data support that Abeta can drive tau pathology and neuronal loss without inherently obvious anatomic colocalization between plaques and areas of neurodegeneration. So the next part, I would like to talk about the neurovascular system. In humans, the brain receives up to 20% of our cardiac output. So if the cerebral blood flow stops, brain function will stop in seconds, and damage to the neuron will occur in minutes. So the normal vascular and neural relationship is extremely critical for normal brain functioning, because every neurons in the human brain has its own capillary. The total length of capillaries is about 400 miles, and the capillary surface area available for molecular transport is about 20 square miles. The length of the brain capillaries is reduced in Alzheimer's disease, and the vascular reduction can, we know, the diminished transport of energy, substance, and nutrient across the blood-brain barrier, and it can reduce the clearance of potential neurotoxins from the brain. So what exactly it looks like, the neurovascular unit? The schematics of this is a capillary cross-section, you can see the tightly sealed endothelium or endothelial cells, you can say, shares a common basement membrane with pericytes. They are called pericyte because they are encircling the endothelial cells, and astrocyte end feed wrapping around the capillary wall. I will show more pictures about it later. The neurovascular unit, as I said, comprises of pericyte, endothelial cells, vascular smooth muscle cell, glial cells, and neurons. This particular unit regulates several functions. Some of them are hemodynamic neurovascular coupling, microvascular permeability, matrix interactions, neurotransmitter inactivation, neurogenic coupling, to say the least. So what is the main mechanism and how it this A beta can eliminate from the brain? First, it's called enzymatic or glial degradation. Activation of the A beta degrading enzymes such as neprilysin, angiotensin converting enzyme, insulin degrading enzyme. And another part is called transcytotic delivery. It inhibits the receptor for advanced glycan end products, or RAGE. It's actually a transcytotic influx of circulating A beta into the brain. And the perivascular drainage is nothing but generated by cerebral arterial pulsations. So malfunction of any of these proteolytic pathway could impair the A beta clearance and can lead to more accumulation of this A beta inside the brain vasculature. So what exactly happened with these A beta and endothelial cells inside the brain? Because as I said, glial phagocytosis, enzymatic degradation, transfer to the CSF. On the A beta peptide can cross the blood-brain barrier because they are very tiny by active transport. And who does it? RAGE actually mediates these transcytosis, to the perivascular place, which is called A beta influx in the brain. Whereas this A beta efflux, which is getting out of the brain to the periphery in the blood, will be mediated by two proteins. We call it lipoprotein receptor P1 and P-glycoprotein. So endothelial mediated mechanism of A beta production. We know that APP and A beta can be observed in the perivascular space of the Alzheimer brain, maybe of endothelial origin, and this A beta induced blood-brain barrier permeability and transmigration of mononuclear cells, vasoactive and apoptotic effects of A beta, reduced cortical cerebral blood flow has been observed in the infusion of A beta 40 in the mouse. The phenomenon is RAGE dependent. That means RAGE is actually causing the influx of A beta inside the brain, and it mediated by upregulation of another proteins called endothelin 1. So as I said, there are two hypotheses, or two hit hypotheses to be precise, available to vascular aspects of Alzheimer's disease. Vascular factors such as diabetes, hypertension, and genetic factors coupled with is called APOE, can lead to cerebrovascular damage. We call it HIT1. And within the A beta independent pathway, cerebrovascular damage leads to the BBB leakage and accumulation of blood-derived neurotoxic molecules. Additionally, blood-brain barrier dysfunction within the A beta pathway, blood-brain barrier dysfunction can also cause disruption of this A beta clearance pathway, and ultimately it accumulates in the brain. And also it can cause a synaptic dysfunction, neural injury, neural degeneration, disrupting structural and functional connectivity, and ultimately dementia ensues. So cerebrovascular dysfunction in Alzheimer's disease, what happens in the cerebrovascular, it diminishes the cerebral blood flow, so which can lead to the microhemorrhage or ischemia in the brain, and blood-brain barrier breakdown in the hippocampus in increase in an age-dependent manner. In a post-mortem Alzheimer's disease brain, we have found fibrinogen, thrombin, plasminogen, IgG, and albumin, which are found in the cortex and hippocampus. Another important aspect is called glucose transporter, because it's the major transporter of glucose across the blood-brain barrier from the blood to the brain. And GLUT1 on the cerebral microvasculature has been reported to be very much reduced in the Alzheimer's disease brain tissue. A beta clearance in a sporadic Alzheimer's disease, faulty A beta clearance thought to be promoting the elevated brain A beta levels. RAGE, as I said, is a major protein, efflux receptor protein, which is transports the A beta from the blood to the brain. Normally accesses very low levels in the normal control human, but it is increased with the age. Cerebral amyloid angiopathy, or CAA, are called mostly vascular plaque, and the basement membrane abnormality has also been pretty much pronounced in the Alzheimer's disease because of the thickening, splitting, tortuosity, and development in abnormal inclusions. As you can see in the left side, the formalin fixed brain slice of a 60-year-old male patient, the huge amount of plaque, and also causes mini stroke or microhemorrhages, and also cause a lot of vascular plaque in the endothelial cells of the vasculature. So not only that, 85% of the Alzheimer's disease patients present with cerebrovascular lesions, primarily CAA, also infarcts, and also microhemorrhages. On the right side, you can see from the brain patient samples, which I did just as normal stain in the FSB, you can see a lot of plaques, the parenchymal plaques, and also in cerebrovasculature. So I was talking about GLUT1, blood-brain barrier GLUT1 expression worsens in Alzheimer's disease, neuropathology, as I said, and it only says that GLUT1 may represent a therapeutic target for Alzheimer's disease, vascular neuronal dysfunction, and degeneration. How did they find it? Berislav-Lokovich group. So they did a combination like, breed with APPPS1, which actually expresses two double transgene, and combined with the normal, but also made a GLUT1 transporter, GLUT1 deficient mice, which showed that the blood-brain barrier permeability is increased, and cortical microvascular length has been reduced, hippocampal microvascular length also reduced. Not only that, they also found that extravasation, which means where the blood-brain barrier is leaky, they found the IgG amount in the brain is way up than the normal counterpart. And also tight junctional protein such as occludin. On the right side, you can see I have labeled the red, occludin, zonooccludens, and claudin. And then these actually has been reduced as well. And on the protein level in the Western blot, also you can see there are claudin, zonooccludens, and the occludin has been reduced. So next up, they also found the synaptic disjunction, means the number of we call it dendritic spines in the hippocampus is reduced in this particular bred mice. Also, they found the NeuN, which is called new, and denotes the number of neuronal cells present in a particular area of the brain, also reduced. Also, you can see the neurofilament number also reduced, and also the size of the brain is reduced compared to their wild-type counterparts. Another aspect that I said in the neurovascular brain is called pericyte. Pericyte is the kind of a cell which encircles the endothelial cell, and they actually have a lot of role in different aspects of the like diabetes and hemodynamics and blood vasculature. And also they found that pericyte deficiency can lead to the development of tau pathology and RD neuronal loss, which is normally present and abnormally absent in the Alzheimer's precursor protein transgenic mice, resulting in a cognitive decline. What they found in the pericyte deficient mice, that number of pericyte also reduced in the brain. They also found the blood vasculature also are inundated with a lot of vascular plaques, and also the neurofilaments. So in this also they found that it can accelerate the CAA or the cerebral amyloid angiopathy, because cerebral amyloidosis is diminishing the clearance of soluble A-beta and 40 and 42 from the interstitial place or the street of the brain. And that means endothelial interactions with the astrocytes become more important, because endothelial cells of the blood-brain barrier are surrounded by perivascular astrocytic infiltrate. Not only that, astrocytes can also modulate the tight junctional proteins, the expression of endothelial transporters, including P-glycoprotein and glucose transporters. As I said in the past slides, like the vascular plaques are predominantly present in the Alzheimer's disease human. So on the right side, on the bottom, you can see a slice. It is called, you can see a vessel which are encircled with the astrocyte plaques and the microglia as well. So next up, I'm going to update a little bit about my present study, which is vascular dysfunction in the brain of two transgenic rodent models, Alzheimer's disease, and the dietary impact and relevance to human Alzheimer's. I studied two different rodent models. One is called cerebral amyloid angiopathy mouse model, because it has DATN-IUA mutation. In a nutshell, it actually develops a lot of vascular plaque around 3 to 4 months, and it becomes more and more among the ages. And it also goes to the different area of the brain, and it also shows a lot of cognitive deficits at the very late stage. The bottom one, which is a transgenic one, which we have the breeding colony to study all these different aspects of Alzheimer's. And this is the background of Fisher. And also it does a lot of tauopathy, gliosis, Frank-neuronal loss, which is pretty much common in human. That's why I call it neuronal loss. And it is a good model for study in Alzheimer pathology. So using the CA mice model, we recently studied and published a paper, which is RAGE. We wanted to study because we said the RAGE is the major influx receptor at the blood-brain barrier level, which transport A-beta from the blood to the brain. Normally expect very low levels at the blood-brain barrier, but increased with age. So the characterizing this neurodysfunction will be the key of our study. We found that in this mice model, we found A-beta, aggregates, microvessels. So this is a study where we isolated the vasculature of the brain, and then we studied the different aspects. So we found A-beta aggregates accumulate in brain microvessels, and we also found P-glycoprotein and the RAGE transporter are dysregulated in these microvessels of these mice. We also try to find out whether tight junctional proteins are impacted. Obviously, occludin expression is decreased quite dramatically in the brain of made microvessels of these mice. And in vitro, also, we wanted to find out how much blood-brain barrier permeability is altered in these endothelial cells of these mice. So in conclusion, we find that, these mice accumulate A-beta aggregate in the microvessels compared to the wild-type mice. These particular mice represent, all sorts of proteins decrease, but more importantly, this P-glycoprotein and the RAGE are the two which are very much decreased compared to their wild-type counterparts. And these mice actually present lower level of tight junctional protein. So we also saw in our rat model, by doing lectin leveling, you can see a lot of ischemia in the vessels and compared to their wild-type counterparts. Another study I am right now doing is called olfactory dysfunction in an Alzheimer's disease model. In the human, you can see in the in the middle one, human olfactory bulb, a lot of tangles been formed. So it is not nothing new that the degeneration. It's like Alzheimer's, Parkinson, a lot of other neurodegeneration models or neurodegeneration diseases. So the loss of dysfunction is prevalent. So we wanted to study the temporally what age it starts and how it progresses in the rat model. We also found degeneration of the neurons by using a specific marker. We call it FluoroJC. It actually goes across. So in the 10 or 11-month transgenic rats, you can see the vascular plaque in the olfactory bulb, and you can also see in the FluoroJC degenerating molecules in different area of the olfactory bulbs. The reason actually we took the hippocampus and piriform cortex because olfactory bulb has connection to the hippocampus and piriform cortex as well. So we also, as I said, in the rat, we are still ongoing. There are different proteins we are studying. These are actually part of olfactory bulb studies. And you can see a lot of different molecules such as the amyloid precursor protein. LC3B is nothing but a sequestosome for the autosomal phagosome pathway. So next question is, how does this correlate with the human olfactory bulb? Since I'm involved in the human brain samples, so we also found that our preliminary data suggests that RAGE, which we found reduced in the mice as well as rat, they are inversely correlated human olfactory bulb as well. While RAGE is progressively increased with the Braak stages, LRP or the lipoprotein protein is decreased. So in conclusion of this particular study, we can say both fibrillary and non-fibrillary amyloid plaques are present in different layers of the olfactory bulb. Maximum amyloid deposition was observed in 24 months of Alzheimer's rats, and the FluoroJC leveling present in those areas of the brain where amyloid plaques are present as well. They're showing both neuronal and axonal degeneration around the plaques. Amyloid precursor protein, oligomeric A-beta also increased over time. P-tau gradually increased at all ages. RAGE, which is also increased over time in the olfactory bulb and in this particular transgenic rat compared to the wild-type part. The study which we undertook is called parasite evaluation, and then there are morphological features in this model, mice model. We found that number of endothelial, number of parasites in this model is reduced compared to their wild-type counterparts. We also try to look at the endothelial cell morphology in the hippocampus and cortex. we found is dramatic reduction. The area fraction occupied by these endothelial cells is reduced dramatically in transgenic, and also their intensity of this endothelial cell in the brain is reduced as well. On the right, on the top, you can see in the human also we can see a lot of vascular plaques. They're different and they're globular in pattern. So, one second. So in the mice model, in the CA model also, we can find a lot of vascular plaques. So not only that activation of microglia around the inflamed endothelial cells, microglia are green in, you can see the RECA is called red endothelial cell antigen, which is red in color. So you can see, and the blue is the plaque. So the association of these is pretty much predominant in the Alzheimer model. Again, astrocytic activation, microglia activation around the plaque, and amyloid plaques in the large vasculature. So next up, I'm going to talk a little bit about the tauopathy or tau tangle formation. What exactly is a tau? Tau is a heterogeneous microtubule associated protein which promotes and stabilizes microtubule assembly, especially in axons. Neurofibrillary tangles are a major hallmark for Alzheimer's disease, and these tangles are a bundle of paired helical filaments composed of hyperphosphorylated tau. If you can, I don't think you can remember the structure, but you can see the big structure. One is the C-terminus and all this, N-terminus. But the question is, some of these areas, which when they get phosphorylated, the neuronal structure gets, different. So pathologically, they are different when that particular, site is being phosphorylated. So such as AT8, which is a antibody which actually targets two different site of the kinase. So in the hippocampus, in the 6.8 months, in a particular model of mice, we call it tauopathy model, you can see two different structures. In the cortex, the morphology is quite different. It's also, we have seen this tau, phosphorylated tau present in the rat model, and it correlated with the human samples. At the bottom, you can see on the left, CP13, which actually represents the serine phosphorylated site of the tau protein, but it mostly presents the pre-tangles, not the ghost tangles we generally see on the right side with the paired helical factor, which actually denotes the kinases like serine and 396 and 404, which actually can show you a lot of ghost tangles as well as flame tangles in the brain. Again, continuing CP13, phosphorylated serine 202, you can see the globose shaped, and 8000 because that is a phosphorylated serine 214. Another one is 88 serine phosphorylated and in 205. So you can see different phosphorylation site can show you different morphologies in the brain. And at the bottom right, you can see the ubiquitination and how much it actually shows you the neurites as well as astrocytic plaques in the human brain. Not only that, TOC1, which is the tau oligomeric complex, which actually recognizes the tau oligomeric complex, and immunoreactivity can be shown quite elevated in Alzheimer's disease brains, which is a conformation-dependent antibody whose epitope is revealed upon dimerization and oligomerization. On the left side of the bottom, you can see normal pattern of tau in a age-matched control. Remember, these patient samples are always age-matched because if suppose you are looking at the 90-year-old Alzheimer patient, if you see the 90-year-old control, you will see there is big difference in terms of tangle or pathology or A-beta. But other than that, there should be or would be a lot of other, baseline level, which is pretty close to a little bit inflamed state as well. So this is another study which we undertook about the plaques and tangles, how they are correlated with the hyperphosphorylated tauopathy in the brain of Alzheimer's disease patient, and also how much it correlates to the gliosis. So inflammation in the Alzheimer's disease brain is pretty much apparent, as you can see in the unaffected control human brain. Microglia, they look pretty normal, and their processes are ramified and they look normal. However, when they have a huge inflammation because of these A-beta as well as tau, a lot of inflammation or rod microglia and also proliferation of these will be obvious along the vasculature, and also in the periphery of the meninges. You can see on the left side of the at the bottom. On the right side of the bottom also you can see a lot of activation as well as a lot of degradation or dystrophy of this microglia. So next few slides we'll be talking about the management or the factors, vascular risk factors, lifestyle and environment. Diabetes is one of the major vascular risk factors contributing to develop AD. Atherosclerosis, as I said in the past, the APOE-induced atherosclerosis or APOE genetic factors can be a big role. Hyperhomocystinemia, beta amyloid deposition shifted to the vasculature, and a group of people are studying about this, showing that if you have hyperhomocystinemia can lead to the vascular plaque in the brain. Traumatic brain injury increase the production of A-beta, and recent studies suggest that in the American national, like we call it NFL or football league, which is pretty rampant, about 110 people have been studied which are dead, and they show that the chronic traumatic encephalopathy is being caused by TBI or repetitive TBI, I would say. Sleep inadequacy and daytime sleepiness can increase the risk of Alzheimer's disease and development of A-beta pathology. Peripheral inflammation, pneumonia, and other causative agents such as H. pylori, HSB have been found in Alzheimer's disease postmortem brain tissue as well. Alcohol and substance abuse. Alcohol can abuse can increase the cerebral blood flow and cause blood pressure and can disrupt the blood-brain barrier, endothelial tight junction phosphorylation, and it also can degrade the gelatinases. We call it matrix metalloproteinase 9 and 2 because those are extremely important protein as well. How to prevent Alzheimer's disease? Restoring the blood-brain barrier transport machinery by using LRP upregulation. PICAM is another protein which I'm rightly working on. It's called phosphoridyl binding clathrin assembly protein. It involves in endocytosis because the internalization of the cell receptors and intracellular trafficking of the endocytic proteins. GLUT1, as I said, GLUT1 is being impacted in this Alzheimer's disease model, blocking the RAGE, microvascular stabilization, MMP9 inhibitors, antihypertensive drugs in the market as well. Lifestyle improvement of vascular health, diet, exercise, and environment enrichment. There are a lot of drugs have been tried, not a single drug yet available or yet been proved to suggest that they can cure Alzheimer's disease. And then there are 3 different drugs been approved just to treat the asymptomatic relief of Alzheimer's disease in related studies, but nothing have been proved yet. So countering this neurovascular impairment with a PPR agonist, acetylcholinesterase inhibitors, and other compounds may improve the clinical outcome of these patients or can delay the progression of this severe dementia. This particular slide, I'll be spending a few minutes because it just appeared in Lancet and their recommendations are extremely important for us. What are the 3 new modifiable risk factors of dementia they have introduced? New evidence support these 3 modifiable risk factors: excessive alcohol consumption, second, head injury, and air pollution. This has been revised from the Lancet 2017. Less education, hypertension, hearing impairment, smoking, obesity, depression, physical inactivity, diabetes, and infrequent social contact. So these are, and now what are the modifying these 12 risk factors might prevent or delay up to 40% of the dementia. Head injury is pretty much rampant among the older people because of the fall. So be ambitious about prevention because prevention is about policy and individuals. Contribution to the risk and mitigation of dementia can begin early and can continue throughout life. So it is never too early or too late. These actions require both public health programs and individually tailored intervention because there are 5 different kinds of dementia, like frontotemporal lobe dementia, dementia with Lewy bodies, Parkinson disease, and then ALS, then Alzheimer's disease. So all these dementia could be caused by different neurodegenerative disorders. So how to maintain systolic blood pressure of 130 millimeter or less in midlife from around 40 years old. Antihypertensive treatment for hypertension is the only way we know effective preventive medication for dementia as well. Encourage use of hearing aids for hearing loss and reduce hearing loss by protection of ears from excessive noise exposure. Reduce the exposure of air pollution and secondhand tobacco smoke, prevent hand injury, limit the alcohol use as alcohol misuse and drinking more than 21 units weekly can increase the risk of dementia. Avoid smoking uptake and support smoking cessation to stop the smoking, as this can reduce the risk of dementia even in the later life. Provide all children with primary and secondary education. There is a very big correlation between education and all these neurodegenerative disorders. Reduce the obesity and the linked condition of diabetes, addressing the putative risk factors for dementia like sleep, though lifestyle intervention will improve our general health as well. So what are the future direction of research one can find or look to? Detection of early biomarkers, CSF or the cerebral spinal fluid, and the blood serum. We have done some work about the blood serum work, which is like exosome studies, or other factors can also be found. So, but the problem with the blood serum, sometimes it is not accurate because of some other reasons. Early detection, loss of smell, which is pretty much preponderant in the neurodegenerative disorders. It is not yet known whether it is a cause or effect. Intestinal neural pathology, a biomarker for identification of Alzheimer's disease. I'm still working on the intestine of the human as well as in the rat studies, where we are trying to find out how much intestinal gut microbiota can tell us regarding the Alzheimer's pathology. Can exosome be used as an early biomarker? I think it can. Gut microbiome, as I said. Can lipid profile? There are a lot of companies that are up to this particular biomarker development. It's not yet done. Why Alzheimer's disease is prevalent in women? There are no clear indication why. The group of studies says estrogen reduction or the postmenopausal can be the cause, but other studies say the estrogen supplemental or HRT is not going to affect or mitigate the risk of Alzheimer's disease. Does chronic stress play in the role of developing AD? Yes, there are a lot of studies going on that intense chronic stress for a long time can also have severe impact on our brain health. Does lack of sleep can potentially expedite the progression of AD? Yes, it does. Endoplasmic reticulum stress reduction can ameliorate AD pathology. There are a lot of studies going on. Recently, NIH gave $50 million to start, some startup companies to develop certain particular drug which can mitigate or cure Alzheimer's disease pathology. And what is the take-home message for my presentation? In the cognitive decline is notable years or even decades after extensive plaque pathology accumulates, as almost always associated with the onset of tau pathology, and neurodegeneration has not been shown to be reversible. This is one of the clinician as well as Alzheimer research scientist, David Holzman from WashU University. And here's my acknowledgement slide, which is our division director, our center director, and my colleagues and collaborators across different part of the US. And I would like to finally acknowledge my human brain research, there's a neurobiobank in NIH, and our study support group. So I would like to have any questions or comments or suggestions from you guys.
Okay, thank you so much, Dr. Sarkar, for such an outstanding and mind-blowing presentation. It was very informative. Definitely, we have lots of questions from our participants.
Sure.
Yeah. So first question is there any involvement of amyloid beta proteins in any other brain pathology?
Amyloid beta production not only it causes the cognitive deficits, it also causes the neuroinflammation, it causes vascular permeability, I would say blood-brain barrier permeability. Because of this A-beta, it actually, as I said, it actually is the root cause, or it can, start other processes. So bottom line is pathology, in terms of neuropathology, it increases the proliferation of microglia, proliferation of astrocyte, proliferation of everything. So still the group of scientists believe that A-beta formation is the key cause of later part or later stuff, which actually we can see in the. So yes, it is A-beta actually can cause a lot of other morphology as well.
Okay, thank you. Next question. Also, is there any initial indication of alternation of blood-brain barrier in Alzheimer's disease?
Early indication? No. There is no such indication in specifically for Alzheimer sporadic population. There is no indication you can find whether the blood-brain barrier is being compromised. Only, as I said, two things: blood-brain barrier can be compromised. Suppose I'll tell you, if you are severe stress, your blood-brain barrier permeability or blood-brain barrier can be a little bit permeable fast. Second most important thing is there are some proteins they are trying to, it's called neurofilament L light chain. They're trying to see whether that actually, if you have more of that particular one in the blood serum, whether that indicates as a early blood-brain barrier permeability. Because for the TBI, like when they get the concussion, specifically for the players who get a lot of concussion, and then what happened, brain will release those, and whether those proteins are being released from the brain in the periphery, and that could be used as a biomarker. So there are studies going on which actually suggest that a lot of biomarkers can be used, but only this is the only one which is FDA approved and said this neurofilament light protein can be be used as a early biomarker for detection or some kind of thing. Yeah, go ahead.
Thank you. Next question. There have been some reports of antioxidants and flavonoids from plant sources being able to arrest apoptosis in the mitochondria and endoplasmic reticulum. Could you shed some light on this?
Yes. So most of the flavonoids, they are polyphenols, right? So plant-derived structures, like I have worked a little bit on the maurine, mangiferin, and then other antioxidants such as TUDCA, trehalose, and all sorts of. So the question is, what we see in the in vitro and what we see in the in vivo is a little bit different, right? So in vitro system, most of the time you are dealing with the monolayers where your cells and everything is pretty much controlled, and you do not have other structures or other proteins associated which you don't see in the in vitro. But on the other hand, brain is a very heterogeneous organ in a sense. Each part of the brain, such as the olfactory system, cortex or hippocampus, will be completely different than the other areas of the brain. So the next thing is, your question is whether these can be used. There are studies which are actually like one of the factors, I think one of them is called curcumin, right? Like which is we found in turmeric, right? So turmeric has a lot of different components. So a lot of them are antioxidant, a lot of them are anti-inflammatory. The question is how much it will penetrate to the brain. Okay? How much you will need to ingest to get to the brain. There is a formula, it's called log BB value and log BB value and log PM. These values represent how much, suppose you take 100 gram of a medicine, how much is going to go to your brain and how much is going to go to the hepatic portal system. So that's why this any chemical which you see in the animal or in the in vitro study, you cannot just directly go to the clinical trial, because first of all, what percentage of those chemical will go to the brain is most important. Okay, otherwise your kidney as well as your liver will be damaged forever. You got my point?
Okay.
Dr. Sarkar?
Sir, I request you to just stop sharing your screen. Your face is
The
not
presentation is going on. So please stop sharing your screen so that the presentation stops and then you can, yes. Okay.
Can you hear now?
Yes, we can hear.
Okay, go ahead. Shirojit?
Yeah. Next question.
Sir, the presentation is probably still on.
Okay, let me stop presenting.
Yeah, stop presenting.
Go ahead.
Yes.
Yeah. What is the role of brain-derived neurotrophic factor and CREB in Alzheimer's disease?
BDNF and CREB, you mean?
Yeah.
Well, there are a lot of studies going on about BDNF was a little bit busted because they tried to use that one for human trial, but it did not go through very well, honestly. So yeah, I mean, the BDNF has a role. BDNF, we always know that brain-derived neurotrophic factors are extremely important for brain health, and they are trying to use that one for not only the Alzheimer's disease, other factors. When but no such effect has been found in the Alzheimer's disease yet.
Okay. Next question. Is there any blood marker which is prevalent in Alzheimer's disease patient?
Yeah, means as I said, it's like inversely proportional. Like if you see amount of A beta present in the brain and the blood, it is inversely proportional. Such as in the beginning of Alzheimer's disease, there are 3 different stages in Alzheimer's disease. We call it BRCA 1 and 2 together, 2, 3 and 4, and 5 and 6. So early stage of BRCA 1 and 2, you see more A beta in the brain and less in the periphery. Okay.
Okay.
Sometimes you can see, and you can sometimes you can see mostly it's in the periphery. You can see more if the patient has no clog or something is not the impairment. But when it become more and more, like in an age group, you can see, like in age as well as the Alzheimer's disease patient staging, you can see more in the brain, specifically in the cortical area. By looking at the MRI and the micro-PET CT, you can see lots of them in the all those areas. So blood biomarker may give you a lot of indication, but it should go with the another thing, which is micro-PET CT as well as MRI to particularly detect those whether they have the Alzheimer's disease or not.
Okay. Thanks a lot.
Sir,
Next
can
question.
you turn on your video, please? Your video is off.
Whose video?
Your sir.
Do you want it? I thought you don't want it.
No, sir.
Someone told me to stop it, right?
No. Yes, sir. It's perfectly all right now.
Next
Go ahead.
question. Yes, next question. Can overactivity of cytokines like interleukins that has been happening in COVID have any effect in causing dementia?
Oh boy. COVID-19 is a different beast. COVID-19 cytokine storm, and I've been reviewing molecular neurobiology about the COVID-19. Only trouble is with the COVID-19, not much is known whether COVID-19. See, COVID-19, the route of like a pathogenic mechanism, right? So people are saying it can go through your eye, like lacrimal gland. It can go through your nose and nose from the nose septum and this area, nasal turbinum, and then it can go to the olfactory system, and from the olfactory system it goes to the brain. That's one mechanism. Another mechanism from the air passage, it can go to the lungs, and from the lungs it goes to the blood, and from the blood it goes to everywhere in the body, right?
Okay.
So your question is whether it can cause dementia or not, right?
Yeah.
So the problem is, I think it will take really a big effort for someone to answer, because Dr. Fauci and others say it about the viral load. He suggested, Dr. Fauci and the big scientists in NIH, he suggested the viral load, amount of viral load you get actually determines your severe disease. So, and another thing, all the other comorbidities, such as someone has specifically diabetes, someone has COPD, someone has, I would say, emphysema, someone has asthma, all these pre-existing conditions, we can aggravate this, particularly COVID-19 situation. So which could be, but later on, we don't know yet, because COVID-19 patients are coming right now. And if it is early enough, and how long do they live? As I said, and if someone is going to the ventilator, and ventilator is the last worst possible case scenario. In those conditions, other organs can be impaired as well because of the shutting down other organs because of the other anesthetics. So anesthetics can also pose a danger to the health as well as the brain health, and also ultimately for dementia. Yes.
Okay. We have another question. Can any viral or bacterial infections which are capable of compromising the blood-brain barrier in a patient also be the reason to trigger Alzheimer's in them at an early stage? What is the likelihood?
First of all, lot of bacteria. Have you heard of bacterial meningitis and viral meningitis, right?
Yeah.
So meningitis is the inflammation of meninges. So bacterial meningitis, that means your blood-brain barrier is leaky and it is inflaming the. So viral meningitis or bacterial meningitis is very, very common in the early childhood because they do not have the blood-brain barrier development. And it's also very much common in the elderly people above 70 or above 80. Now, your question is, say, viral. Yes, if you're immunocompromised person, if you're taking immunocompromised drugs or something, if you have autoimmune disorders, they are more likely, but there is no study yet have found that autoimmune disease patient can have Alzheimer's disease, because of this. But again, Alzheimer's disease to me is a kind of a ultimate, like effect or something. There's a difference between the senile dementia and Alzheimer's dementia. To be honest, senile dementia people or the patient will know that the reason of using the phone, or someone calls you, that person will know the reason of using the phone. He may not remember who is calling, but if in the Alzheimer's patients people, like if you give the phone, that person will not know what is the functionality of this phone. So that's the basic difference. But coming back to your point about the viral, and yes, if the viral load, if the bacterial load is huge and is actually causing irreversible damage to the blood-brain barrier, yes, it can ultimately cause. That's what been found in Alzheimer's disease patient brain. They found huge amount of other viral as well as bacterial stuff in the brain.
Okay. Now, lastly, I have two questions. So one is whether Alzheimer's disease is very much controlled by different kinds of environment or not. Whether environment plays a crucial role or not to control Alzheimer's disease.
As the Lancet study suggests, air pollution, as they're saying, air pollution, smoke and smog, and other can contribute a big part of this, dementia. I mean, they are saying dementia specifically. See, in the Parkinson disease, for example, if you are born and brought up in an environment where you have too much of manganese in your local areas where you can consume a lot of manganese, we call it hemi-Parkinsonism, can cause. So the same way, we call it environmental, gene, as well as body interaction. So yes, environmental play a big role, but you cannot just say the environmental can cause Alzheimer's disease.
Okay. Last question: whether it is age dependent or age independent?
It is age dependent. Obviously, after 65, it jumps. The chance of you having Alzheimer's disease ups and ups and up. But only for familial disease, it is very early age because they have a typical mutation, very aggressive form of mutation, and they go between 37 if it is 14, chromosome 14, if it is more than if it is 21 or other, and it will be 45 to 55. But for other sporadic people, like anyone in the world, it will be based on their lifestyle, habit, and other factors. Yes.
Okay. Sincere thanks, Dr. Sarkar, for such an beautiful explanation of all those questions. Okay. So now for today's webinar, our second speaker is Professor Ashish Kumar Panigrahi, but he is just about to join. I hope everybody can understand he is so busy person. So he's just about to join actually. Just please wait and be patient for a few seconds.
No problem. Yeah, I'll be back within 10 minutes. I'll take a break.
Okay, it's absolutely fine. Yeah.
ওটা অন করে নিব
No. No. এখানে অন করছি না মনে হয়।
আমি তো অন করেছিলাম, তুই কি প্রেস করলে আবার? দেখি সুইচ না, অন তো আছে না, চার্জ সবই আছে, চার্জ করো।
হ্যালো,
Yes, sir, you are audible.
হ্যাঁ হ্যাঁ, now I am audible.
Yes, sir.
Hello.
Yes, sir, you are audible.
Okay, then I can start because I am very late. Just now I entered my house, and a very good evening to everybody. First of all, I convey my heartful gratitude to beloved principal of the college and the organizers, Rajit, Sur, and other member of the organizing committee for waiting for me for long for some minutes because I was on the way returning from Bardhaman University and the road was very jam today. So I welcome all the participants and the distinguished guests from USA. I heard his lecture on my returning time. He delivered very nice lecture, and now I will go for my lecture. And the first slide lecture is I am just showing, please. Hello. No, এটা sound off আছে. You can.
Yes, sir.
So now I can start.
Yes, sir.
Okay. Thank you. Excuse me for my late. So today in the webinar on the recent advances in biological science, in that relation I have selected one topic, that is the advancement technologies in aquaculture. What are the advancements have been made for the development of aquaculture? So we know what is aquaculture. What is aquaculture? Aquaculture is the culture of aquatic organisms such as fish, crustaceans, mollusks, and aquatic plants. Aquaculture involves cultivating freshwater and saltwater population under controlled conditions. Control condition means that is the culture will be made by human beings and can be contrasted with the commercial fishing, so that the culture practice will be so much so that the fishers, that is the person involved in the culture practice, they can earn some money by selling their products, that is in the commercial basis. So the objectives of this presentation is that we can first
Sir,
show.
may I stop you? May I stop for a few seconds? Beg your pardon. Actually, I just want to give some kind of introductory part actually. Okay. So just beg your pardon. Just please, sir, wait, please.
It
Okay.
is required now.
It's just wait. Please wait. Just wait.
Oh.
Now for today's webinar, our second speaker is Professor Ashish Kumar Panigrai, Pro Vice Chancellor, University of Burdwan, West Bengal, India. Professor Panigrai did his PhD from University of Kolkata in Fishery Science and Ecotoxicology in the year of 1988. He has an enormous amount of experience in teaching as well as in research, almost about 35 years. Teaching experience 20 years approximately in University of Animal and Fishery Science and University of Kolkata. And also service experience 12 years as Fishery Technical Officer, Planning and Developmental Cell, West Bengal Government of India, and research for 3 years another. He held the post of professor from 2013. He has got lots of prestigious awards. Few of them are Clean Environment Education and Promotion Award 2018, Swami Vivekananda Excellence Award in the year of 2017, Bharat Shikha Ratna Award in the year of 2016, and lastly in the year of 2020, he has got Eminent Teacher Awards. And he worked as a head of the Department of Zoology under University of Kolkata from 2012 to 2014 and 2018 to 1st of June 2020. Then he joined as Pro-Vice Chancellor in the University of Burdwan from 2nd of June onwards. And he is currently absorbed as Pro-VC. And under his guidance, 16 actually awarded, 2 theses already, and also he has published 115 papers and already communicated, and he has published book 6, contributed chapters in different books 12, and article published 40. And also he has been administrative experience, a huge amount of experience also, and has been life members of several society. Some of them are Society for Science and Environment, Bhopal, JDSI, Bodh Association of Aquaculturists, Bhubaneswar, etc. And he has presented lots of papers and lectures in different kinds of conference, both national, international, approximately 60. So, and his areas of research is actually fisheries and aquaculture, aquatic and industrial pollution, biodiversity and conservation, and ecotoxicology. Okay, now I would like to request Professor Panigrai to deliver his lecture. Over to you, sir. Sir, please start your lecture.
Thank you. Thank you. So again, I have to start from the initial. That is this topic of my today's lecture. That the advanced technologies in aquaculture, because we know aquaculture is a very prominent subject now, and it is related with the livelihood. Many people depends on aquaculture. So the basic definition of the aquaculture is the culture of aquatic organisms such as fish, crustaceans, mollusks, and aquatic plants. And aquaculture involves cultivating freshwater as well as saltwater populations, that is the marine populations, under controlled conditions. What do you mean by controlled conditions? Means that is cultured by the human beings, cultured by persons with controlled conditions, so that the conditions which are required for the culturing of the aquatic animals, both from the freshwater or from the marine water, that should be controlled by the culturist who culture the different animals and plants in that medium, and can be contrast with the commercial fishing. That is, it is very much matching with the commercial fishing. So the productions, what are the produced in from this culture, they must be in commercial basis. That is, huge production is involved in this aquaculture practices. So the main objectives of the this present topic is we have to know what is the status of the aquaculture in the world fish production. How world fish production at present, the situation of the world fish production, and the world fish food systems. What are the systems involved for the production of the fish as a fish food organisms? Then how much fish do we produce today? And we have to know how much fish we produce today, and what is our demand actually. How much fish do we expect to produce in future? Because our demand is gradually increasing with the increasing of the populations. So we have to take a target to meet up the goal, and how much expect to produce in future, and how much fish do we need to produce in future, what is the actual demand of fish in near future. So implications from the developing country perspectives. So what are the implementations of these programs and its perspectives in different countries, and what are the challenges? There are three main challenges I will discuss later on, and the research agenda to meet these challenges. What are the research agendas have been taken throughout the world to meet up these three challenges, which if we meet up the challenges, then we can go for production of huge number of fishes, and we can also achieve our target. So research agenda to meet these challenges. What are the research agenda at present have been taken throughout the world to meet the challenges, and reflection on the skills we need, and what are the reflection, what are the skill is needed to achieve this goal. So this is the main objectives or main theme in my lecture. So now I can go for why we need to modernizations the aquaculture, why we shall go for modernization of the aquaculture. That is to develop through research and training sound technical base for organized growth of aquaculture in the region, both as small scale and as large scale and commercial enterprises. So we can go for small scale aquaculture systems. If we have less water bodies, then we can go for small scale aquaculture system. If we have large water bodies or water bodies given by the governments, like different entrepreneurs will come out to meet up these productions. So we can go for small scale aquaculture practices, or we can go for large scale aquaculture practices, and large scale aquaculture practices is very much related with the commercial enterprises. Next, to increase food production in the form of fin fish or shellfish in the region. So we can go for different type of fish production. We have two types of fish production. One is the fin fish, that is the freshwater fishes, and the other fishes which have the fins in their characters, in their body, and or scale fishes or shellfishes. Shellfishes means the body is covered with shell, that is the prawn, crabs and lobsters. These are the shellfishes, and the other fishes which have the fins in their body, they are called fin fishes. So these are the two types of fishes group. We can go for culture to meet up our food product, meet up our protein, animal protein, to promote rural employment. If we go for large scale aquaculture practices, then we can promote rural employment, because in the rural sectors there are so many small ponds or water bodies which are laying vacant without any scientific culture practice. If we promote the aquaculture, is one of the sector in to the unemployment youth, for the unemployment youth, then we can promote rural employment by engaging them in different mode of aquaculture practices, so that the different ponds or water bodies which are laying vacant in the rural areas, these can be utilized properly for the production of the different type of fishes. Then to make aquaculture a strong medium, not only for saving but also earning foreign exchange. So this, if we go for large scale aquaculture, then we not only earn some money for ourselves, for our livelihood, but also the excess production we can go for exporting, and in lieu of that we get foreign money, that is a dollar. So what are the development goal in this process? So to improve the availability and access to financial resources for aquaculture investment, to reduce the social and economic impacts of aquatic animal diseases. Because if we go for aquaculture practices, aquaculture productions, then there is very sensitive cases that anytime the large scale fish culture, the fish can be infected by different type of diseases. So we have to give emphasis to control the disease. That is, we have to go for management practices. We have to take care during the cultural practices to make efficient use of land and water resources, because the land and water resources should be efficiently used in a plan wise, scientific plan wise, that water body should be used so that we can get more productions. To improve the environmental management and image of aquaculture, that is, improve the environmental management. So the aquaculture must be eco-friendly, that do not make any hazardous effect on the environment. So it is scientifically that should be oriented so that the pollutional load or environmental hazardous should be checked during the practices. Then to create an enabling policy environment for sustainable aquaculture. So some policies should be adopted so that this type of aquaculture practices should be environmentally eco-friendly, what I have already told. And the practices should be sustainable, means there is no consequences in checking of the production. The production will be gradually in increasing trend, and the problems which can produce some pollutional impact on the environment, that should be checked. Then to promote responsible development of aquaculture through a progressive legal framework. So this should be some law we have to carry out, or we have to go for bindings of some laws so that the legal framework should be followed for the safety of the environment as well as the safety of the fishers who are engaged in different type of aquaculture in the marine water. Because every time in marine water there is every possibility to face accident or any type of disasters by the farmers or by the fishers who are engaged in these practices. To improve the flow of knowledge so that the farmers who are interested in such type of aquaculture practices, they should be trained properly so that they can improve their knowledge in the practicing processes. To improve the human resource base to manage and to operate aquaculture development, so we can employ here the human resources properly. We can use the human resources for their in this practice so that they can go for aquaculture development. To maintain and enhance social harmony, and it is also one type of things that social harmony should be maintained properly. To increase participation of farmers associations and the private sector in aquaculture development. If they think through these practices they can earn more dollars, so they will come and they will adopt these aquaculture practices. To move towards a market-based system, the culture should be in such a way that should be heavy demand in the market. So the market system should be followed. What are the demand in the local area where the practice we can continue? So it should be market oriented and the system should be market based. To improve quality and image of the aquaculture product, quality should be maintained, because without better quality or best quality, nobody will touch the products. So quality controls is the main principle to produce all the things, and we can export these things through quality controls. So the aquaculture is a sector of fisheries which have gained great importance over the years. In 2000, if we go for a history, in 2014, fish harvested from aquaculture amounted to 73.8 million tons in 2014, with an estimated fast scale value of US dollar 160.2 billions. That is, what are the types? That is the finfish, and how many millions ton? 49.8 million tons. What is the value? 99.2 billion dollars. Then mollusks. These are the aquaculture products. Mollusks, that is the 16.1 million ton, and its value is 19 billion. Crustaceans, that is the crabs and other things, the 6.9, the production 6.9 million tons, and the value is in the US dollar, that is 36.2 billion. It accounted for 44.1% of the total fish production of the world. So 44.1% of the total fish production of the world is in the process. So this is the fish matter. So how the increase from 1965 to 2005, how it is gradually increase the productions in comparison to other animal proteins. 20% animal protein intake for 3 billion people. Then an important part of the healthy diet, because animal protein is a healthy diet, nutrients essential to cognitive and physical development, especially in children, because in children we need the animal protein, so their cellular development and their growth. Then most of the dependent on fish are among the most food and nutrition increases. That is the, what are the comparison? This I have seen here, and the commodity is the beef, eggs. What are the animal proteins? That is the beef productions. That is Oceania gradually increasing there. Similarly, eggs, 16 eggs, then fish 16.5% in Oceania region. Then the mutton and the goat, it is also increased. Then offal, pig, poultry. These are the animal proteins, and they are the region, that is the Oceania, North and Central America, South America, Europe, China, Asia, excluding China, 40.4%, then Africa, and the total world. So these are the food supply chain per kg per hectare. Then the world fish production. What is the trend of the world fish productions in million tons? That is the marine, brackish, and freshwater. Because we have three types of fish production, we can get fish from three sources. One is the marine sources, that is from oceans, from the seas, and from brackish water, where the river is mixing with the sea, that is the brackish water, and the fresh water, totally fresh water, that is the sweet water from rivers or from reservoirs or from ponds. So this is the inland fisheries, and total catch is 62 million metric ton, that is a 41% in 2011. And total wild catch means from the marine sources, that is not for, we not go for culture, that is wild available, and that is catch is 94 million metric ton, that is a 59%. Out of 100%, 41% from the aquaculture practices, that is from freshwater, and 94 million metric ton, that is a 59% from the marine waters, as per the source from the FAO. Then the world fish production and food use consumption. How the consumption is related with the projection, that is the aquaculture in China from 1976 to 2030. The projected is 2030 is the target hour. So aquaculture in China, then aquaculture in world excluding China, then capture fisheries. What do you mean by capture fisheries? Capture fisheries means that is from collected from the wild. That is, we not go for any culture, we only go for capture. We can catch the fishes, but we not go for culture of the fishes. That is the capture fisheries. And another sector is the culture fisheries, that is aquaculture, where we go for culture of the fishes or aquatic organisms including plants. So capture fisheries, world excluding China, that is the rate gradually increasing, and the food use of the world, that is the last, that is also I have seen in this graph. So now we can go for Indian scenario. What is the actual scenario in India? So India have come a long way in terms of the aquaculture technology and boosting its productions. India has come long way after very much struggle. After 1950, India go for aquaculture practices. Before that time, it was a traditional practices in the rural sectors, mostly in rural sector. Not they have adopted any scientific culture practices. So various agencies have dedicated themselves in developing newer methods for enhancement of the culture fisheries. That is various agencies, they come forward because they understand that if we go for aquaculture practices, then our earning will be more than of the agriculture practices, because we have already proved that if we go for culture of fishes in 1 hectare pond in comparison to agriculture practices in 1 hectare land, then the production will be nearly 3 times in case of fish production than that of the agricultural productions. So the many new entrepreneurs are developed and they come forward to adopt the new technology, the scientific culture method of aquaculture, and they start for the enhancement of the culture of fisheries, that is the culture of fishes. And government organization, the government also have taken separate ministry, that is the animal and fishery department, fishery animal and fishery ministry, and the ministry established after independence different organizations, central organizations for the research and development of the fisheries in India. And the most prominent institutes are CIFA, Central Institute of Freshwater Aquaculture. They are involved for the culture practice technology development in freshwater aquaculture, that is in riverine fisheries, in pond fisheries, in wetland fisheries. They involve how these rivers, these wetlands, these reservoirs can be developed with new technology for the fisheries culture. Then SIVA. SIVA is the Central Institute of Brackish Water Aquaculture. That is, they are very much concentrated with the brackish water aquaculture. Means brackish water means mixing of marine water and the fresh water. That is the river mouth where the river enters in the ocean. That portion is called the brackish water system. In brackish water, where there is a mixing of the fresh water as well as the marine water, so this area is very much productive. Different type of fish can be cultured, and these fishes are having very high market value in comparison to totally freshwater fishes like rohu, katla, mrigal. Similarly, another institute is the Rajiv Gandhi Center for Aquaculture, very advanced center. After the death of Rajiv Gandhi, the center has been initiated by the central government, and there advanced aquaculture researches are going on. Then the National Center for Sustainable Aquaculture Systems. There are so many other aquaculture organizations, like in our West Bengal, there is CIPRI, that is the Central Inland Fisheries Research Institute in Barakpur, and we have in Wetland Fisheries Research Institute at Salt Lake, and we have so many research organizations in Chennai, Central, that is the Marine Fishery Research Institute, in Kakdeep, that is the brackish water fishery research institute. So there are so many institutes they are involved to find out different type of new technologies which are involved in the aquaculture practices. Then top 5 fishery state in India. That we have so many states, but these top fishery states are Andhra Pradesh, that is their inland productions, that is lakh tons, 28.45 ton inland productions in the year, that is the 2011-12, and marine production is the 6.05 lakh tons. Total production is the 34.5 lakh tons in the year 2011-12, and the in case of in rupees, it is nearly 16 crores rupees, in crores, 16 crores rupees and 59 lakhs. Then the West Bengal is the second position in production in comparison to Andhra Pradesh, that is 15.57 in inland fisheries, that is the freshwater fisheries. Then marine fisheries 1.85, total 17.45. In 2012-13, it is 18.85 crores. Then the Gujarat in third position in fish productions in India, that is 1.34 inland fisheries, freshwater fisheries, and marine is more, that is 7.01 highest, and total 8.35. And they are in 2013 and 14 and 15, that is 33.4 crores. In Kerala, it is 5.34 inland water, and the marine water is 1.51, and total 6.85 2015-16, and it is nearly 30 lakhs, 30.4 lakhs. And Tamil Nadu, that is 1.85 lakh tons in inland water fisheries. Then marine is the 4.97, total 6.8, and they are 37 lakhs 87,000. So all India production total is 89 lakh tons, and they are out of that marine is 35.88, and the total is the 1.25 125.9, and it is in 17-18, and total is the 45 crores rupees, 45,000 crores rupees. So the strategies for the development of aquaculture in India, what are the strategies have been taken by the scientists? So to extend the farming areas by using the land and leasing policy and revival of the defunct or old farms. There are so many old farms, so we have to take the land requisition, have to take, and leasing system also we have to go for leasing system of different ponds. Then the scale up the production potential candidate varieties of aquaculture. So what are the potential fishes they can go for culture? We have to select the candidate species who can go for culture, and these are the sea bass for mariculture or for marine culture of fishes, that is the sea bass, then mud crab, then tilapia, then pampano and cobia. These are the fishes they are cultured in the marine systems, but tilapia is cultured both in marine, in brackish and freshwater. So to increase area under the production, only 13% of the suitable land brought under aquaculture. So out of the 100%, we have only achieved 13% suitable land for scientific aquaculture practices. Tie up the private or public hatcheries shall be done to have multi-species hatchery cycles, because we have to go for production of the seeds and we have to build many hatcheries. So many private public sectors or private parties, they come forward to establish hatcheries in different parts of the India for production of the fish seed as per our demand. To increase the hatchery production of high quality seed for all species farmed at present. So now at present we have so many hatcheries, both government, private and public sectors, and they are engaged in production of high quality seeds of different species as per our demand. So how much are we likely to produce by 2030? In 2015 it is 75 million metric tons, but we have to produce, if we achieve our target, then we have to produce 109 million metric tons. That is a 2.5% average increase is required as per FAO, and total, that is the 75 is this production in 2015, and 61, then 136 total, that is the fresh water and marine water, total 136 in 2015. But if we go to our target of 2030, then we have to go for production of marine 109 and fresh 61, total 170 million metric ton fish should we need in 2030. So estimated fish requirement, that is the total demand. If we go for estimate to fish for all, then our total demand is 2038. But we need, at least we need 2028. And our requirement, if we go for average, then in 2030 our requirement is the 232 million metric ton. Can you imagine how we can go for production of fish for direct human consumptions as per the increasing populations? So expectation, our expectation is we will be 62 million metric ton short unless we do something. See, if you show the chart, that if we go for scientific pisciculture till in 2030, in the year 2030, we will be shortfall of 62 million metric tons. Then we have to go for more scientific way of culture, to adopt scientific way of culture to meet up this demand. So aquaculture, this is the aquaculture growth. That is, aquaculture will need to produce 39 million more tons than the 109 currently projected. This will be require a shift from an expected 2.5% to 4.7%. We accepted 2.5% increase, but we actually need 4.7% to meet up our demand. So we cannot ignore fisheries, and we need to reduce waste and loss. So we have to also take precaution on the waste and the loss during the aquaculture practices, because in scientific aquaculture practices, in extensive aquaculture practices, if we do not go for good management, then we have to face a great loss. Just in 1990, we faced a great loss in prawn culture throughout India, because we started the supra intensive culture of prawn in India, which is not environmentally suitable for India. It may be possible in Thailand or Japan or others foreign countries, but in Indian conditions, our soil, our water, our environment is not matching with that of the other countries, because here if we go for supra intensive culture, that is the production will be more than 60,000 kg per hectare per crop, and in a year we can produce 2 crops. So it is very much difficult, and the environmental pollution come, and all the seeds died in the farm, and Indian people who are engaged in prawn culture, they suffer a great loss. And from that time, government has banned the supra intensive culture in aquaculture in India, and they have give the advisory to go for extensive or semi-intensive or maximum intensive culture. The production challenges. What are the production challenges? How do we produce enough fish to meet future demand and need? These are the in Kerala, they are going for marine fish culture. Then sustainability of the challenges. What are the sustainability required? That is the digging of the ponds. How we can go for mass culture of fishes in big water bodies? How do we ensure that the fish food system in both economically and ecologically sustainable? So we have to ensure, we have to come forward. So distribution of challenges. What are the challenges? How do we ensure that the fish is affordable and available for all, and that the economic and the nutritional benefit from the sector are equally shared? So we can, if we go for aquaculture, then we have to think about the economic value, because the livelihood depends on the production. If the production is more, then livelihood will be increased. If the production is less, then the livelihood, they cannot achieve their livelihood, so they will not get interest in aquaculture. And the nutritional benefit, so such type of fishes should be cultured which have the nutritional benefit for us, so that we can take the fishes in our daily meals. So interlink of the challenges. What are the interlinks? The production challenges which are related with that one, that is the sustainability challenges, and the distribution challenges. We have 3 challenges. One is the production challenges, that is we have to go for highest production, and the sustainability challenges, that we have to carry out gradually in the increasing trend. The production will be in the increasing trend, not in the decreasing trend. And we have to also take care about our environment. So the culture should be eco-friendly. Similarly, the distribution challenge. If our production is more but there is no market, then we will face a loss, because we cannot sell our products. So the distribution challenge is also, that is a challenge. So we have to ensure the market where our produce will be sold in, so that we can earn the money. So technologies for the sustainable intensification in aquaculture, sustainable of the fish production system, developing nutrition sensitive fish value chains, and improving aquatic agricultural livelihoods. So these are the interlinked challenges. So technologies for sustainable intensification of aquaculture. What are the research challenges? That is the polyculture with small indigenous species. We can go for polyculture with small indigenous species, because small indigenous fish species have the heavy market value now. So finding technology and the financial combination that accelerate the pro-pore aquaculture development, sustaining rates of genetic improvement of fish suited for poor farmers, and improving the nutritional value of farmed fish. Nutritional value should be improved, so that human being can purchase the fishes for their day-to-day use in their meal. Developing sustainable feed also, alternative feed also we have to develop, so that we can meet up the proper growth of the fishes. So techniques used in the mobilization of the aquaculture. What are the techniques already used in aquaculture? That is the egg development and spawning should be the promptly in many species by injection of the hormone extract from the pituitary gland, because induced breeding practice has been introduced in the year 1956 in India, and its father is Dr. Hiralal Chaudhary, who injected first to go for induced breeding by using pituitary gland. And there is a short period in the early life of the aquatic animals, mainly fish, when their sex is not fully determined. Feeding them with a hormone treated feed can push them to develop for male characteristics. Some fishes, some techniques have been developed to change for sex so that male fish can go for development more faster than the female. And aquatic animals, in case of shrimps, do not readily mate in captivity, are artificially inseminated by extracting the sperm capsule from the male and attaching it to a female that has ripe ovaries to replicate the natural mating process. These are the different process for the breeding. In the time of the feeding, particular species of microscopic algae are collected from the sea or freshwater or grown in the tanks with just to right mixture of the nutrient make them flourish. So that is the algal culture should be needed, and aqua seed should be cultured, feed should be cultured, so that the plant should be cultured for their feed. Then there are different type of synthetic hormone discovered and used in the induced breeding. These are the HCG, that is human chorionic gonadotrophin, is a glycoprotein hormones produced by the placenta in the pregnant woman. HCG, human chorionic gonadotrophin. It is also given in the fish for their productions of seed. Then ovaprim. Salmon gonadotrophin is a very important drug in place of the pituitary extract produced by the salmon gonadotrophin RH and doperidone. These are the all synthetic hormones applied in induced breeding. Then primogide. It is a dopamine antagonist having ovulatory role, that is evolution, it helps evolution, role of luteinizing hormone RH on the brackish water fish like Mugil cephalus and Latus calcarifers. Then DOC A, that is the 2-desoxycorticosterone acetate. It is a very effective drug for Clarias species and Heteronesis fossilis, that is the cat species, magur and singi. These are the injection process. The fish is injected as per their body weight, because we know as per the body weight the dose is fixed, and here the dosage of induced breeding, that is the first dose, and we have to give 2 doses, first dose and second dose, and female are given 2 doses and male given only 1 dose at the time of second dose of the female. So female dose per kg body weight is a 2 to 3 ml per kg body weight, and in the second case it is double, that is a 6 to 8 mg per kg body weight, and for male the first dose of the female, that is a 2 to 3 ml per kg body weight during induced breeding. So then after injection the fishes are then given to the breeding pool for their pairing, so that they can release eggs. These are the breeding pool where the male females are introduced at the ratio of 1:2, 2 female 1 male. Then a rearing pond where after that is after the release of eggs, the eggs are taken to the hatchery. These are the Chinese hatchery, very famous hatchery now. Chinese hatchery where 90% seed produced within 3 to 5 days, and then the seeds, that is the spawn, were transferred to the rearing pond for rearing fingerling. Then the fingerlings are transferred to the stocking pond to produce table fish that is above 500 grams. So different types of aquaculture. What are the types of aquaculture? That is, I have already told you that the extensive aquaculture, traditional aquaculture, then semi-intensive aquaculture, then intensive aquaculture, and this intensive aquaculture again divided into flow through method, that is the raceway culture, and another is the recirculatory culture. So we will go for one by one. That is the intensive fish culture. Intensive fish farming system is the oil managed form of fish farming. That is oil managed, that is scientific managed. And to achieve maximum production in intensive fish culture, maximum production of fish from a minimum quantity of water. And this system involves small ponds, tanks, raceways with very high stocking density, that is 10 to 50 fish per cubic meter of water. And that is the flow through the method raceway culture. The raceway culture is defined as the raising of fish in running water. That is running water. Water is always in the flowing way, like river, like fountain. Raceways are the designed to provide a flow through system to enable rearing of much denser population of fish. Because we know if water flows from continuously flowing, then the atmospheric oxygen will be caught by the water, then the dissolved oxygen level will be increased, and so high density fish can be cultured in this that is flow system, that is the raceway culture of aquaculture. And the recirculatory system of aquaculture means a recirculating aquarium system, that is RAS, can be defined as an aquaculture system that incorporates the treatment and use of water with less than 10% of total water volume replaced per day. Every day, that is the 10% of the water volume will be replaced in this process. That is the circulatory tank, and this circulatory tank to solid filtration. That is filtration, that is the solid will be filtered, then the biofiltrations, then carbon dioxide will be removed, then pumping system, water will enter, then disinfection, that is the all the germs will be killed, then the heating and cooling system in this system, so that the fine particles will be removed and the pH will be controlled and oxygen will be increased. Then that water will be again circulated in the tank and fish will be cultured in the circular tank. This is the systems of the recirculating type of aquaculture. This is the RAS system. Then there is a very famous system, that is the in India it is very much suitable in India and China, that is the integrated aquaculture techniques. What do you mean by integrated aquaculture technique? Integrated fish farming is nothing but integrated fish farming referred to the production, integrated management, comprehensive use of aquaculture, agriculture and livestock. That is, it is a admixture of the aquaculture, agriculture and livestock. And it is a history that about 1500 years ago India adopted this type of integrated aquaculture, but it is that time it was not scientific. Now it has been shaped in the scientific ways, and to 1620 fish livestock farming is first seen in China. Then in the 50s of the 19th century, integrated fish farming practices were transferred from China to Japan. First it is initiated in China, then to Japan, then Russia in 1850, and Madagascar in 1914, then India in 2000 in 1930 onwards, that is traditional culture. Then in 1960 or 1970 onwards, it will be given scientific mode and scientific culture. And what are the types of integrated fish farming? Agri-fish system. In the agri-fish systems, it includes paddy, that is the paddy cum fish culture. It is called paddy cum fish culture. That is, in a paddy field, we can go for culture of fish also, along with the paddy. Then horticulture. Horticulture cum fish culture. Because in horticulture garden, we can go for culture of fishes. In the tank, fish will be cultured, and in the embankment, different type of horticulture can be initiated. So the embankment will be utilized properly, the soil of the embankment will be utilized properly for production of different type of horticulture produced, and in the pond, that is in the water body, fish will be cultured, and the water will be utilized in the horticulture systems, and from the horticulture system the waste will be utilized as a food of the fish. So it is called horticulture cum fish culture. Then the mushroom cum fish culture. With the mushroom, the culture can be done. Then the sericulture with fish culture. Sericulture will be done in the land, and fish culture will be done in the pond, and the embankment is fully utilized through sericulture. Then vermicompost. Vermicompost is also done in the land, in the embankment, and the fish will be cultured in the pond. And vermicompost, that is the vermicompost, that is the fertilizer that is used, that will be used in the fish pond, so that fish can grow very faster, and their water or their waste will be utilized for the vermicomposting. And similarly, it is the agri cum fish system. Similarly, livestock cum fish culture, that is the with cattle cum fish culture, pig cum fish culture, goat cum fish culture, poultry cum fish culture, duck cum fish culture, rabbit cum fish culture. So these are the systems. That is, with the livestock we can go for culture system. With the pig, we have to make, I am showing here, that is the actually scientific drawbacks, the scientific picture. That is the fish will be in the center, and there is the duck cum fish culture, and that is the cow cum fish culture, that is the pig cum fish culture, then the horticulture cum fish culture, then the poultry cum fish culture. So these are the systems which called integrated fish culture. That is, in one water body and their embankment fully utilized by different cultivation systems. And this is the picture of the integrated chicken cum fish culture or poultry cum fish culture. We can go for, we can keep this in this shed, the poultry birds, and the poultry birds, they can drop their droplets in the water, and their droplets, their dung, poultry dung, is rich with the nitrogen, phosphorus, and these are the fertilizers. They can enhance the water quality parameters. That is, they can enhance, they can fertilize the water bodies, and so that the water color will be blue. That is the appearance of the phytoplankton and zooplankton, which are the primary fish food organisms, are the primary food of fishes. So fish will get the food from their dungs, and the fish will grow. There is no need of giving fertilizers, additional fertilizers from outside in this integrated type of farming system. Integrated of pig, duck, fish, vegetables systems. In a one pond we can go for duck cum fish culture, we can go for pig cum fish culture, we can go for vegetables cum fish culture. And these are the vegetables, they plant, different type of plant, banana cum fish culture, guava cum fish culture, papaya cum fish culture. These are the plants which are grown on the embankment, and the ducks shed in the embankment side. The duck dropping will fall in the pond to enrich with different type of fertilizers, and they enhance the water quality parameters for growing of the primary food of the fish, that is the phytoplankton and zooplanktons, and pig also. In this system, based on use of the pond, which not only meet needs to pigs, but also enables fish and ducks to be kept, water is also useful for vegetable productions. This water can be used for vegetable productions. So then another thing is the cryopreservations of fish gametes. These are the very scientific new methods. That is the cryopreservation. What is cryopreservation? Cryopreservation is the long-term preservations of biological materials. Then fish technology, that is the gametes, embryos. We can preserve the embryos, gametes, at ultra low temperature, usually minus 196 degrees Celsius of the temperature in liquid nitrogen. So we can preserve the fish gametes for long time, and at the time of our demand, we can collect the gametes and we can go for production of the seeds. At this temperature, the cellular viability can be stored in a genetically stable form and is affected only by breaking down the radiations. And why the cryopreservation? Because at a time we cannot get the fish, that is the gametes. So during that time we can use this gamete through by taking from our preservations, and we can go for induced breeding, and we can also go for production of androgenetic fish. These are the first requirement of the cryopreservations, and this is the overview of the cryopreservation systems, how we can go for cryopreservations. These are the pictures. Then we can go for use of synthetic carotenoids in ornamental fishes, because we know ornamental fish culture is a very prominent that is the for employment generation in the rural sectors. This ornamental fish have demand throughout the world as a pet fish, and we can enhance their color by using synthetic carotenoids. How these carotenoids are used, that is the carotenoids belonging to the family of the fat soluble pigments and have various sources, including the natural sources, that is the fungi, yeast, and the algae. Animal sources are the crustaceans, and the synthetic sources are the lycopenes, then genophanes and astraxin and the beta carotenes. So these are the carotenoids which are used in ornamental fish coloration, enhancement of the ornamental fish coloration, so that the fish will be looked very beautiful and colorful and eye-catching. So these are the synthetic carotenoids now available in the market. So importance of the fish carotenoids in the fish diet. So that fish carotenoid is used in the fish diet. There are three main groups of the pigments are responsible for the coloration of the fishes. These are the pteridines, then melanins, and the carotenoids. Among them, pteridines and melanins are produced endogenously, but carotenoids cannot synthesize endogenously, so it must be obtained through diet. So through diet we add carotenoid for better coloration of the ornamental fishes. And this is one type of another scientific age, that is the biofloc. In the biofloc, this is the big containers, and these big containers we can go for culture of different type of fishes. That is biofloc technology is a wastewater treatment system. So যাকে বলে ময়লা জলে মাছ চাষ, so wastewater treatment system we can use biofloc. It plays a vital role in aquaculture. Biofloc technology helps fish to grow healthier and quantity. This technology is also balancing nitrogen and carbon in water, because nitrogen and carbon balancing is the main things to get the water more fertile. If carbon is less, then the water will be fertile. If the nitrogen is in balancing conditions, then the water will be more fertile. This technology was developed by Varem Evinementhan in Israel. It is Israel technology. That is the biofloc. Then biofloc technology, low investment of rupees only 30,000 only for the installation of the 4-meter dia tank, and production of 600 kgs of biomass within 5 months, and it within one harvest, and the suitable for the Indian tropical conditions, because we are a tropical country. The production we can go for biofloc. All types of fisheries can be cultured in biofloc. And scheme for the biofloc fish culture system. This is the schematic diagram of biofloc culture systems in pond systems. So aquapods, another type of terminology, the aquapods, that is the most advanced type of aquaculture. Aquapods is There is a culture in cages were inputted in earth, oceans farms operating back in 2011. They are constructed with a series of triangular net panels, that is the pen culture, fashioned into a sphere, invented by ocean farm technology, now known as the Innova Sea system. A drive team and biologists monitor the cages, which are suited for open ocean conditions and diversity of species. Different type of fishes can be cultured in the ocean. It is just like a ball, and the that is the aquapod system, and the pot is inserted in the water, and the fishes are seen inside the water. These are within a cage, so fishes are cultured within a cage in the floating system in the sea. And these are the floating fish farming system. Another type is the floating type of. These are floating type of ponds we can build in the ocean for culture of fishes, different type of fish species through different cages. And sustaining the fish production system, research challenges. These are the research challenges we have to take, otherwise we cannot achieve our goal. And nutritional sensitive value of the chain. These are the nutritional sensitive. And enhancing aquaculture systems. So we have to go for the aquatic as well as agricultural system, because the India is the agricultural rich country. So we have to we have to produce agricultural products. Otherwise, we cannot get our food which are needed for us, along with the fish as a animal protein. So lastly, I can say globally the technological innovations are showing a positive impact on aquaculture, diversification, success, investment potential, and international technology exchange. Biotechnology is a useful tool in developing disease-free strains of fish seed, highly nutritive fish food, etc. Newer innovations in aquaculture system designs are paving a way for tremendous growth in production in future. Offshore culture systems are to be promoted along with the land-based culture systems for enhancing production of marine species, and catches from the wild are also required. In India, the aquaculture has shown growth over the years, but has more scope in improving its present status by adopting new technologies. Government agencies alone are conducting research and technology transfer in the field. Lack of entrepreneurship is a constant factor in India culture of fishery industry, because entrepreneurship are they are not interested in fish culture like that of other industry. So private collaboration may help the industry in gaining the capital investment for technological development and growth of aquaculture in the world. So this is the end of my lecture. Thank you to you all for having patience to listen to my lecture, because it is long durations, and it's going to be 10:30, and I am also very tired. Throughout the day I engage different type of activities. Thank you to you all. Thank you.
Thank you so much, sir. I can understand, and you are very much tired. And so much again for giving us so just very few questions basically asked you. One is, how significant is the involvement of microflora such as bacteria and algae in fish culture ecosystem?
Yes, this that is a microflora is very important in fish culture system, because if with the microflora we can go for culture of now mass culture of microflora and other planktons for the fish feed preparations, and also to make disease free of fish by using different type of microorganisms.
What are the possible eco-friendly management technologies in aquaculture during this pandemic?
I am not listening. Some disturbance in the mic. Please.
Can you hear me, sir?
Say.
What are the possible eco-friendly management technologies in aquaculture during this pandemic?
That is, during this pandemic, most of the farmers are remain there. They are in their home, and but one thing is that due to lockdown, most of the industries are totally stopped, and their production is totally stopped, or partially they are running their industries now, and so the discharge of different type of industrial effluents is very less, and the aquatic environment is lesser polluted due to minimum discharge of the effluents or waste from different sources like municipality or other sources. So there is every possibility of growth of fishes during this pandemic period if farmers take some special care.
Okay, sir. Thanks a lot for your kind explanation. And sincere thanks to both of our invited speakers. Now we would like to conclude this webinar with vote of thanks that will be delivered by Stuti Das Mohapatra, Faculty Department of Zoology. Over to you, Stuti. Can you hear me? Stuti?
Yes, I can hear you. Surajit, I can hear you. Am I audible?
Yeah.
So I'll now proceed with the vote of thanks. So thank you to both of our speakers and everyone else who is listening now. We deem it a great honor to propose the vote of thanks to all those who have helped us in making the webinar such a resounding success. First of all, we would like to thank both of our respected speakers for gracing today's webinar. Thank you so much, sir, for your very interesting and thought-provoking presentation. We would like to express our profound gratitude to our respected Principal Ma'am, Dr. Kinjalkini Bishwas, for her constant support and enthusiasm. In addition, sincere thanks to the convener, Dr. Surojit Shur, HOD, Department of Zoology, for his guidance and arrangements of this webinar. We would like to thank our staff members of ICT cell, especially Srimati Sunetra Sengupta and Sri Sanjay Mojumdar, and all members of the organizing committee who have made this webinar a successful event. Finally, we are very much thankful to all the enthusiastic participants who turned up in such great numbers and made this webinar successful. Once again, thank you all for your kind cooperation. Over to you, Surajit.
Thank you, Stuti. So the recorded version of this webinar will be available in our YouTube channel, and please submit the feedback form only after the official conclusion of the webinar, and please do not hurry. All will get respective e-certificates at the earliest. So goodbye to all of you and have a nice time. Hope soon meet up. Okay. So goodbye to all of you.
Thank you. Good night to everybody.
Yes, sir. Good night.
Thanks to have a patience to listen to my lecture. Thank you.
Thanks a lot.
Okay, thanks a lot to both of you.
Thank you.